Hi, everybody, once again. It's great to see you all here for another special iCow webinar. I'm looking at the numbers and I'm thinking that I should tell you a story and stall for time a little bit. But I don't have a very good story to share with you, other than where I found our special speaker. The SDGs are extremely dear to my heart. That's the Sustainable Development Goals. And nobody's ever challenged them. Nobody has even questioned them. Everybody takes them for granted. And the fact of the matter is, we're so far off achieving them that they are like the big white elephant in the rooms of many conferences and many forums. But they're there and they act as a benchmark for some of the things that we need to do to stay within the planetary boundaries as a human species, but we're not doing a good job of it. I was listening to a presentation where somebody spoke about the SDGs in their entirety and questioned how we were ever going to achieve them if we didn't focus on the microbiomes in those. And that immediately caught my attention. And I was like, who is this individual? Let me listen in closer. And he is our guest speaker today. I'm not going to introduce him right now. I'm pushing for time and giving you a bit of a story as to why. And for those that come in later, then they're going to miss the story, which is now over. So now we're going to go to, we've hit the number of about 30, it's time to start. For those of us that are here for the first time, I am Sue Kahumbu. I'm an ardent regenerative farmer. I'm the CEO of Green Dreams Tech Limited, where my team and I power iCow, which is an e-extension platform focused on regenerative agriculture. Team iCow and I, and there are many of them in the house today, we optimize smallholder farming practices in Africa through the iCow platform and ecosystems using our apps, our online trainings, our iCow soil advocates, that's you guys, webinars, and radio. I'm also a Soil Food Web certified lab technician, and I give a big shout out today to all the Soil Food Webbers in the house, those will be joining, and all the iCow soil advocates across the continent. Our current class has students from seven different nations in Africa, which is great. Before we start, I'd like to hand over to fellow Soil Food Webber in South Africa, Wesley Soul, who's joining us once again to manage our Q&A during today's session. So over to you, Wes.
Hi, Sue. Thanks for the introduction. Yeah, as a soil food webber here in the Western Cape, I've spent a lot of time looking at Africa's microbiome and helping clients, whether they be growing grapes, rooibos, all different kinds of fruits and veg, basically understand their soil a bit better through the soil food web techniques. So testing all the types of microorganisms that you find in your soil and helping them to really optimize their processes as well, from small scale up to large scale. So I'm very excited today to help out in this talk and listen to Thulani and just understand our microbiomes a bit better, because there's a lot of studies going on around the world and Africa is unfortunately not a focus. So I think Africa has to focus in on itself, and I'm excited to be part of the process. And thanks very much, Sue, for the opportunity and iCow team behind the scenes. And yeah, I don't want to hold up anymore and let you introduce Thulani. Thanks, Sue.
Thank you for that, Wes. So the iCow webinars are designed to bring cutting-edge science to our audience of farmers, citizen scientists, and just curious individuals from across the continent and beyond. And I'm delighted to introduce our speaker today from our own continent. So I want you all to sit back and get ready to be blown away. Thulani Makhalanyane is a professor in the Department of Microbiology and the School for Data Science and Computational Thinking at Stellenbosch University. He earned his PhD at the University of the Western Cape in 2013, and his research focuses on understanding the ecology of microbial communities in extreme environments. He's co-authored over 85 publications in leading journals, including Nature, Science, Proceedings of the National Academy of Sciences, and the International Society for Microbial Ecology, ISME. Thulani serves on several editorial boards and currently serves as editor-in-chief for reviews and perspectives of the ISME Journal and senior editor at M Systems. He also serves on national and international panels representing South Africa in bilateral discussions. Thulani was elected to the board of the ISME Society in October 2018. He is the director of ISME's Ambassador Program and is a member of the Executive Advisory Board. Phew! Phew! Closer to home, and for the soil geeks in the audience, just a couple of days ago on March 25th, Thulani authored an article in the Daily Maverick called Microbial Guardians of Marion Island, unveiling the hidden world in subantarctic soils amidst climate change challenges. We love how soil microbes are finally getting the attention they require in climate discussions. Thulani, we are indeed extremely honored to have you with us today. The floor is yours. Over to you.
Thank you very much, Sue. Thanks for the invitation and the generous introduction. So it feels like I've known you for a while, but we've only just met. But already I'm quite enthused by your energy and your commitment. And I look forward to working with you. So thanks again for this invite. I'm going to talk about Africa's microbiomes, but I'll take a more general perspective in talking about microbiomes and try and convince you why African microbiomes in particular matter. I'll talk a little bit about soil, but I'll use our studies based on looking at the microbiomes of African individuals as an example of one of the studies that we've done in the continent. As Sue mentioned, I work a lot in extreme environments and looking at microbiomes from a range of different ecosystems. But the main reason for this is that although we live on this planet, what we don't often realize is that this is actually a microbial planet. All aspects of every ecosystem on Earth are underpinned by microbial communities. Whether you look at the oceans, whether you look at the air, whether you look at this, the soil below us, and most of you working on soil microbiomes would of course have a good appreciation of this, is that microorganisms form the basal step of all food webs and consequently control all ecosystems. So what we know is that microbes mediate a variety of ecosystem services. Microbes play an important role in human health. They help us digest complex. Sorry, there's a bit of background audio. They help us digest complex metabolites, including things like cellulose that our bodies cannot break down. Microbes in the environment, including in the oceans, play an important role taking up carbon dioxide. For instance, our studies in the Southern Ocean have shown the important role of microorganisms in carbon sequestration in marine ecosystems, such as the Southern Ocean. We know that microbes are also important in terms of their impacts on climate change, because there are several studies that have shown now that microbes could represent potential feedback mechanisms which could further exacerbate the effects of climate change. Those of you working on soil know the enormous potential that soil microbiomes hold by means of regenerative agriculture and the potential of using soil microbiomes as a mechanism to sequester and take up a high proportion of carbon dioxide that we release through anthropogenic activities. So the microbiome is increasingly becoming an important area of research. So in the last few years, in the past decade or so, the One Health concept has been defined as recognizing the importance and connectivity between plants, animals, and humans. I propose, and we feel, that microbes actually are an underestimated component of the One Health concept because they essentially underpin all aspects of these connections between plants, animals, and humans. Microbes, of course, link soil organisms through things like antimicrobial resistance genes to plants and to animals. Microbes are important in terms of allowing plants to take up, for instance, nitrogen through their capacity to fix nitrogen directly from the air, providing it to soils, which of course then are important in terms of agriculture and become the food that we as humans consume. So microbes are actually important across all ecosystem services. But I feel that the work that's been done so far has tended to exclude microbes in looking at their role, for instance, in the Sustainable Development Goals, as Sue has mentioned, or in the connection between ourselves and the environment around us. In particular, one area that's been quite understudied is the role of African microbiomes. And very few studies have actually looked at the diversity and function of African microbiomes across a range of different niches. So these are demographic projections by continent by the year 2100. You've all probably seen this plot. You've all been told how Africa has the youngest population, how Africa is growing in terms of its size. But one of the things that stand out for me when I look at this plot is the enormous opportunity that this growth holds for us as Africans. There's opportunities in terms of the economic growth, the social development, and also an opportunity to sustainably use ecosystems around us in a way that has long-term sustainable impacts on all of us. So by leveraging the opportunities that are offered by the microbiome in terms of addressing the challenges that we all currently have, we as Africans can actually unlock our full potential across all of these spheres that I've mentioned, including the economic and social spheres. But when we look at the work or the current knowledge that's been generated from African microbiomes, what you can see is that from this figure that's shown here, which essentially represents all the genomic data that's been deposited on important repositories such as the NCBI in the US, is that the vast majority of genomic data actually come from the global north, and very few studies have focused on African microbiomes. When you look at just a basic search on Scopus and other citation databases, you'll see that well over 100,000 publications have been authored on microbiomes to date. When you put in a search criteria to restrict it to African studies, it's just over 512 papers that have been published on African microbiomes. 100,000 and 500 really kind of makes it very crystal clear the large gap that we have in terms of understanding the microbiomes of African systems. And I'll tell you a little bit more about why some of these studies are needed and why they're important. The few studies that have been done on African microbiomes have tended to focus on gut microbiomes. And I'll give you an example as well from our own studies that have looked at African microbiomes. The studies that have been published to date, what you'll see here is that there's a disproportionate amount of sequence data that's from Tanzania, principally looking at the Hausa population in Tanzania and using them as a sort of indicator of what urban or pre-modernized lifestyles look like, and comparing these mostly to individuals in North America. When you look at other systems, including South Africa, there's relatively substantially fewer studies that have focused on the gut microbiomes of South African individuals and also across all other countries that have been studied. So these are the latest data sets that include studies on human gut microbiomes. When you look at the size of Africa, when you consider the populations and the enormous potential of studying African microbiomes, and in particular gut microbiomes, there is an important need to increase the number of studies that have focused on our microbiomes. So about a year ago, a couple of us met and we proposed a roadmap for expanding microbiome research on the African continent. As part of this roadmap, we called on essentially 3 measures. The first is coordinating research networks. The second is putting forward specific policy interventions. And the third includes infrastructure development to allow a microbiome research. Under the first sphere, one of the things that we envisage doing is essentially trying to have a representative researcher focused on all aspects of the microbiome, including soil microbiomes, across the African continent. We've been using some of the networks that we have through the International Society for Microbial Ecology, where I direct the ambassador program, to identify researchers in each country. So far, I think we have about 15 countries on the continent that could serve as representatives and ambassadors for microbiome research in each of those countries. If you know, or if you yourself are interested in joining this network, please drop me an email. The second is that we're calling for specific policy interventions. One such policy intervention, we've started, and there will be a paper that's published hopefully in the next couple of months, that essentially outlines the roadmap or framework for how we as African researchers should collaborate on microbiome studies with researchers from the global north. In that, we call for things like more equitable distribution of resources, having research that's done not in a colonial style, but is actually driven by Africans, where we take the forefront in terms of designing and implementing studies that focus on African microbiomes going forward. The third aspect of what we've been working on is to specifically establish infrastructure across each of the 4 quadrants of our continent. By this, what we mean is we recognize how expensive it is to in terms of generating microbiome data sets, right? It can cost at least about $1,500 to generate a shotgun metagenomic read. By leveraging and centralizing infrastructure for genomic sequencing, we hope to start negotiating with international companies on how we can reduce the cost of generating microbiome data across the African continent. So this is just a picture we took at our first meeting in Côte d'Ivoire, where we met as part of this consortium in terms of finalizing how we as Africans can start working together towards recruiting and obtaining international grants towards that are aimed at focusing research on African microbiomes across a number of different research priority areas. And we've identified essentially 4 different priority areas. The first focuses on conservation. The second is on biodiversity. The third is on agriculture, and I won't speak about the efforts aimed at sustainable agriculture. I'm pretty sure you guys have you've got better expertise in terms of this area than I could have. And then the fourth, it focuses on looking at the health implications of studying African gut microbiomes. As part of this study, we essentially are trying to establish discrete research priority programs that focus on elucidating various aspects of African microbiomes. For example, the work that we do on conservation essentially tries to study a range of African ecosystems that are currently undisturbed, to measure and determine how, for instance, introduction to mining and other economic measures might impact on all of those ecosystems. Because of how microbes and their metabolic mechanisms respond more rapidly to shifts, microbes are important sentinels of measuring ecosystem change before it even happens. So we're trying to establish through these efforts several sentinel sites across the African continent that will provide long-term data focus on measuring shifts in the microbiome. And there are equally other studies across the different spheres that I've outlined. So I believe, and many of us believe, that Africa has enormous and untapped potential in looking at our microbiomes. We know, for instance, this map shows the distribution of soil orders using established nomenclature. And what you can see is that there's a diversity of different soils that are present throughout this continent. Not studying the microbiomes of these systems mean that there is potentially a high diversity of potentially uncharacterized and novel microbial communities across the African continent. Of course, the scale of these efforts requires considerable investment in terms of providing the genomic sequencing that would be required to establish at least a baseline data set of the diverse nature of African microbiomes, and in this case, soil microbiomes. Again, I want to link up to something I showed earlier where I talked about the One Health concept, and that is the connectivity between terrestrial, aquatic, and marine, and also host-associated microbiomes. By studying soil microbiomes, we essentially can understand, for instance, the reservoir of antimicrobial resistance genes that may be harbored within those soils, and then tend to understand more fully how recruitment of these microbes can impact host-associated microbiomes, including us as humans, and potentially that has links towards understanding how terrestrial movement, for instance, in soils, impacts on the aquatic resources around us. So the point I want to make with this slide is that we as humans are connected to the environments around us, and studying the microbiome is key towards disentangling the intricate or the mechanisms of these connections. So some of these ideas that I mentioned are captured in this review article or perspective comment article that we co-authored last year. And as I mentioned, in a few weeks, we hope, another paper will follow on from this paper where we outline exactly how equitable collaborations with African scholars should be governed, so that these are not interactions that are led from the global north, but are in fact driven and led by African scholars. So there are 5 reasons why African microbiomes matter. The first is that Africa, as everyone knows, is a biodiversity hotspot. The picture in the background is one I took quite recently of the mountains in Mpumalanga. But you know, as much as you can see such massive systems, I can show you equivalently other pictures where you can see much more depauperate ecosystems, such as the Kalahari or the desert in Namibia. So all of these systems select for different microbial communities. The microbial communities in these systems also potentially harbor novel genetic mechanisms and novel enzymes that could be harnessed for biotechnological importance. So the second reason is that ecosystem health and functions is also underpinned by the by the microbiomes. Microorganisms play critical roles in terms of maintaining the health and ecological function of different ecosystems. I mentioned earlier how microbes contribute towards nutrient recycling, soil fertility, and plant health. But microbes are also important in terms of maintaining water quality. So it's important to study African microbiomes because it essentially underpins health across different ecosystems. Microbiomes are also crucial for human health and disease. We know, of course, that there's been lots of studies that have shown the emergence of different pathogens. We've not lived through, but we all are living under the COVID-19 pandemic, and that has outlined the important nature of microbiomes, and in particular viruses, as elements that can essentially control human health and also the spread of disease. We urgently need to understand the diverse nature of microbiomes across the African continent because, linked to the next point, with the advent of climate change, that could lead potentially to the development of potentially new viruses and also new pathogens that may together impact on us as humans, but also on environments around us and the capacity, for instance, of soils to provide essential ecosystem services to plants, which has, of course, profound effects on human health. The fifth reason why it's important to study African microbiomes is that microbiomes are important for sustainable development, and also they're quite important for innovation. So looking at the potential of Africa's microbiomes is important, as I said earlier, in not only tapping into the natural resources, but by studying microbiomes, we could essentially scale and pace our development in such a way that it is not destructive to the environment around us, and that will essentially allow for longer term and more sustainable development, which of course will rely on us being quite innovative in how we apply microbiomes across different systems. I'm pretty sure a lot of you working in the regenerative agriculture space know how application of certain consortia of microorganisms not only leads to improved yields across different types of agricultural systems, but that could increase, for instance, the per square meter productivity across the African continent, allowing us to feed this ever-increasing population within our continent, but do it in a sustainable way that does not necessarily harm the environment. So these are the 5 reasons why I think it's important to study African microbiomes and why African microbiomes in particular matter. So our research group at the Stellenbosch University essentially has about 5 different thematic areas. I won't talk about them in great detail, but the first thematic area essentially focuses on African microbiome discovery and innovation. And under that theme, we've got a program where we are looking at the dynamics of African microbiomes. We've got programs where we focus on climate change, effects on African microbiomes. We focus on using microbiomes as sentinels for measuring ecosystem change. So the third thematic area focuses on microbiome environmental surveillance across a variety of ecosystems. One study that we've recently implemented is a study that aims to look at wastewater treatment plants across the continent. We've seen during the COVID pandemic how measuring wastewater was essentially an important parameter of helping to determine or plan for the emergence of increases in COVID infections. So by doing this and targeting a higher proportion of genes that may be linked, for instance, to antimicrobial resistance, that would allow us to use microbes as sentinels or tools for environmental surveillance across the continent. As part of this project, we've essentially started working with about 10 partners across the continent, and we're in the process now of developing a research program proposal, which we hope to submit for funding to fund these long-term studies. The fourth area, linked to African microbiome discovery and innovation, focuses on microbiomes and health. I've recently joined Stellenbosch University, as Sue mentioned, and part of the work that we do through the Center for Epidemic Response and Innovation is to look at how microbiomes are important in human health. We've got a study, for example, where we are looking at how growth stunting in African children may be regulated by the inability of their gut consortia to digest certain metabolic genes, for instance. So I'll quickly run through one example. I hope I'm still doing okay for time, and give you some of the insights that we've garnered from studies where we've looked at the gut microbiomes of African individuals. So there's been a few studies which have looked at African gut microbiomes. I put here an example of a study that was published, I think, a couple of years ago, where they've looked at short and long read metagenomics of urban and rural South Africans. And what they've discovered from the studies is that the gut microbiome of African individuals do not necessarily conform to the simple non-Western axis that has been described quite popularly in the literature. The reason why this Western, non-Western matrix exists is that the studies that have been done so far tend to compare populations that have vastly different genetics and also vastly different diets across compare them to, for instance, populations in the United States. And that tends to obscure the real patterns that you would see by comparing specifically African individuals. One of the key findings from this study is that due to differences in diets, it is quite vital to expand the studies that focus specifically on African individuals across different regions within the continent. So there's NMDS, or non-matrix multidimensional scaling model plot that shows here. You can see that there's substantial overlap between so-called urban and non-urban individuals, as indicated by this map. And you can see also those differences as evidenced by the Shannon Diversity Index that's shown here. So what we did is that we set up a study as well, where we wanted to understand differences in the microbiomes of healthy South African individuals. We collected stool samples from individuals in Venda and compared them to those from an urban location. We had about 100 participants that we could recruit into the study. And what I'll show you is some of the results from the amplicon sequencing based on 16S analysis. And the results for the ITS sequence data have been published, and you can find the papers, or just contact me if you're interested in that. And I'll also show you some of the insights that we've been able to garner from shotgun sequences. So the shotgun metagenomic sequences essentially give you information regarding the functional signatures of the microbiomes within this environment. And what do we find? So again, what we find is that when you look at this slide, is that you'll see the structural patterns that are based on the ordination plots that are shown here, indicate that the clusters are based on rural and urban populations, and that it shows that there are discrete differences, especially when you look at the dispersions across each of the different groups. In the second plot, we essentially look at what they call biomarkers, and you can see clear evidence of different biomarkers when you compare individuals from urban locations to those from rural locations. The bottom plots essentially shows the abundances, or how abundant different bacteria are in urban and rural locations. And what you can see here, without really looking at the details, is that there are very clear differences in the abundances of certain bacteria in the blue in rural individuals compared to the urban individuals. But overall, what you see is that the rural individuals have a much more diverse gut microbiota, and diversity is often associated with a healthier microbiome, because if you have a diverse system, it's more able to withstand any kind of changes, for instance, the introduction of a pathogen, because it's much more resilient to change. So this plot here essentially shows, again, the differences in diversity between the rural and urban groups. But we also used network analysis. So network analysis, as evidenced or shown here, essentially tries to look at the correlations between different bacteria within the guts of rural and urban individuals. So without understanding the exact analysis, what you can see is that when you look at the rural populations, there's a lot more connections within the gut microbiota of these individuals. When you look at the urban individuals, you see a lot more discrete groups. So the connections between the gut microbiome or the connectivity between the gut microbiome on the reliance on each other is much less, right? So if you have a system where there's a lot more cooperativity, again, that system is much more able to withstand any kind of shift that may be introduced, for instance, by acquisition of a pathogen. So it's much more healthy in terms of resisting any kind of attack. So what we also did, because the results that I've shown you before, essentially look at using, based on using amplicon signatures. What we wanted to know is what exactly those connections that I showed you are, and what are the exact, what's the exact taxonomy of the groups that are involved in these interactions. So for this, we use shotgun metagenomics, and without giving you all of the details, we were able to reconstruct a diverse consortia of high and medium quality genomes from these individuals. What you will see is that the genomes that we retrieved are roughly similar, but when you look at certain groups, they tend to be more abundant and absent in urban populations versus rural populations. This group in particular, and Verrucomicrobia, were present in urban populations. The Spirocheta are known to be causative agent of several diseases, including Lyme disease. So their presence in certain groups, for instance, can indicate that in the rural populations where we recovered this from, that there might be a lot more Lyme disease that's present within these individuals versus urban individuals. So then what we wanted to understand from these genomes that we reconstructed is, what does it mean to have a much more diverse gut microbiota versus a less diverse gut microbiota? So from the reconstructed genomes, we then use the genomes, to essentially look at antimicrobial resistance genes. The figure here shows the diversity of urban and rural gut microbiomes. And what you can see is that especially when you look at this plot, that rural microbiomes tend to be dominated principally by two groups, the Bacteroidata and the Firmicutes, and that there's a lot more microbiota in the urban group that harbor genes linked to antimicrobial resistance. So when you look at some of the putative antimicrobial resistance genes, you see a lot more genes that are implicated in antimicrobial resistance in the urban individuals versus the rural individuals. Again, this signifies a state of unhealthy gut microbiome in these individuals compared to the gut microbiota of these rural individuals that we investigated. So just to briefly summarize the results from the study that I've talked about in the past few slides, when you look at previous studies, there are clear differences between high income and traditional communities, and our data suggests that there are also clear differences between urban and rural Africans, again underlying the need to expand studies that focus specifically on African individuals and their variability. Urban and rural South Africans tend to harbor similar gut microbiota. And when you look at the co-occurrence, those networks that I showed you earlier, they suggest quite clear differences in the stability of rural gut microbiota compared to the stability of individuals from urban locations. So these differences may also explain the considerable differences between the gut microbiota of urban and rural individuals across the different samples that we looked at. With this study, what I'm hoping to show you, and irrespective of the systems that you look at, is that often there are trends that are made from studying populations in the global north. But when you compare those trends to rural individuals, and these trends can also expand to results that have been found, for instance, in soil, they often do not hold, and we see differences not only in terms of the composition of the gut microbiota, but also in terms of the diversity of organisms in African niches. Again, this makes the point of why it's important to study the gut microbiota and the ecosystems of African individuals. With that, hopefully, I still have left a bit of time for questions. I'll thank the collaborators that are shown on this slide and end off by encouraging you all to attend the ISME meeting, which will be held in Cape Town from the 18th to the 23rd of August this year. We've just been reviewing some of the abstracts, and we're still open for poster abstracts. We have just over 1,800 abstracts and are looking to welcome microbiome scientists from around the world. There's a dedicated session that focuses specifically on agricultural systems and agricultural microbiomes. So if this is in your line of interest, I especially encourage you to attend this meeting. Thank you very much for listening.
Wow. I think that we should all give you, first of all, a massive round of applause. So we can use our reaction buttons to do that. Hesbon, are you on the screen? There we go. Can we all give Thulani a huge round of applause?
Thank you very much, and thanks for coming. I realize having a talk a day before Good Friday is a lot to ask. Thanks for coming.
It's a good talk. It's a good talk to have before we celebrate much, because these things should continue to be in the discussions that we're all having now. When you have talks in the week and then you get back to work, things tend to get lost in the mix. But now people have time to go home and talk and share because this is being recorded. A couple of things. First of all, congratulations on bringing together the entire ecosystem and working on bringing that continental network together. That's really, really good stuff. So congratulations on that. I have a few observations that I made along the way, and I don't know where to start, but I'm going to start with, I think, this bit here. You said that Africa can can unlock its potential by understanding, working with, and incorporating its microbiomes in decision-making for economic as well as ecological, et cetera, development. What if we don't?
Well, if we don't, and if we follow the economic trajectory, that's been, an example that's been shown to us, we risk repeating some of the mistakes that have been done by others, right? The world is at a point now that we're not living sustainably. We are producing far too much CO2. There's a potential that if we continue on the same trajectory, we're not even risking. We are at a point where we will pass the ecological tipping point, and that has disasters, maybe not for us, but for generations that follow directly after us. And I think if you look at the fact that we are the youngest population, I think we have enormous potential in terms of doing things slightly differently and much more sustainably. And that in itself holds a lot of power and promise.
Absolutely, I agree with you. I couldn't agree with you more. I just hope that we have the political will, and that we can. I think it's all about knowledge, and that's why I actually like doing these webinars, because once this knowledge is out there and it's put in such a way that everybody can access it, people will start thinking differently and making different decisions. Because in the absence of that, we're making the same mistakes. Business is continuing as usual. Our political leaders are continuing as usual. And purely through a not knowing what else to do. So hopefully we create some really different roadmaps and models to follow. The other thing that I thought of while you were speaking was it sounded like when you said you were using the microbiome for environmental surveillance and looking at the differences in the changes with climate change and mapping those, and then looking at those changes. Microbes are the new canaries, basically.
Indeed, I mean, and especially from any kind of system that you look at, the benefit of microbes is that because they've got such versatile metabolisms, if there's any kind of shift in the ecosystem, you can more clearly and more quickly see changes in microbial consortia. So they become quite useful for looking at even small, minute changes within the different ecosystems. So we've initiated studies now of the coast of Durban in South Africa, and just by looking at temporal sampling across different points, you can easily see how, for instance, there's a release of wastewater into the ocean. You can see the quick change in terms of the microbiomes across that system, and they become much easier or more rapid sentinels of changes in those ecosystems. And that has important applications in terms of managing those systems and in terms of long-term ecological monitoring.
Absolutely. And closer to home for all of us who are doing soils and regenerative ag, we see that in our composts, because we actually purposefully drive different changes with the biomes within the substrates. We create an environment for the thermophilic organisms to come in and catalyze the heat and et cetera. And then when the systems cool down, and when you're looking at it under a microscope, you see that change, you see the shift. And you get to a point where if you're looking in a sample and you don't know where it's from, you can recognize the microbes in there. And you can actually say, this is from a sample that was, still being digested. This is from a vermiculture sample. This was from a compost sample that had was, mature or not. And I know that there may be some questions in the group there, and we're going to come to that. But that leads me to: did I hear an open invitation to the soil, the soil technicians that are using microscopes, that are collecting data, to work with you in some way? We're citizen scientists. We may not be doing the genomic stuff, but I think there is still some value in the data.
Yeah. Yeah, there is definitely a value. So one of the points I wanted to make is that, of course, it's not an either or situation, right? I mean, especially when you're looking at microscopy, you tend to be more focused on the most abundant microbes within your particular sample. The value of the genomics is that you can also look at the low abundance taxa, and sometimes these can also be important players within ecosystems, right? Because they might provide some metabolites that are useful across the system. And so detecting their changes is also important. So combining the classical methods with the new genomic tools, which I admit are not readily available to most people, but that's why we're trying to work towards making them a lot more available to everyone. Hopefully, sometime in the future, everyone will be able to take a soil sample, send it to the nearest lab in Kenya and get a profile of all the microbes within the system, and do that as easily as you do with physical-chemical analysis of the same soil sample.
Yeah, and even better than seeing what's in there is notice what's missing, because what we see, what the gaps are what are important now, aren't they? Because many of our soils have been so badly used through our agricultural practices that there's the gap, the biomes are missing, they're damaged. Yeah, okay, so we have two questions in the chat. One says, thank you, Professor Thulani, for this African-focused research work on our biomes. Would you propose from the work already done that Africa could be having some beneficial edge relative to other continents not yet tapped?
Beneficial edge, I don't know, tell me if I'm sort of getting the essence of your question. But if you mean by beneficial edge, I would say, okay, if we look at the gut microbiomes, for instance, right, as one example, you can see that our gut microbiomes are a lot more diverse than the gut microbiomes of individuals from the global north, right? This has a lot of potential if you're looking at things like probiotics, and developing probiotic-based therapies. You want to look for them in African individuals, right, because of the diversity. I can predict similarly by looking at African soils, because of the just varied nature of the soil topology, you're likely to harbor or have soils that harbor distinct microorganisms within those soils. And that in itself is an untapped potential in case you want to look at potential enzymes and functions from these soils. So in that way, I would say we would have a distinctive edge that comes about as from our unique biodiversity. But I might have totally misinterpreted your question. But in that case, I would say yes.
I think you were spot on understanding the question and even your answers there. But it poses another question. When we look at the gut biome, and there is a question there that says, do you have a few suggestions how we can increase the diversity of our gut microbes? I want you to answer that, coupled with, we've all heard about fecal transplants. We've heard about individuals who are very ill and they have a microbial inoculation of their digestive, of their gut, basically, from somebody else who has a healthy gut biome. So my question that adds on to the first one is, do you think we'll be doing that for soils? Do you think we will be moving some soils to other places to inoculate damaged environments? And let's go back to the original question that somebody did ask. Sorry for making this very convoluted and confusing, but it just sparks the ADHD in me. Let's go to the first question was, do you have a few suggestions how we can increase the diversity of our gut microbes?
So if we think about, I guess in this case, soil microbes, there's been lots of studies that have shown that if you do what are called reciprocal soil experiments, where you take soils that might be more diverse and transplant them, that actually seeds a much more diverse consortia of microorganisms in that new environment, and that translates to better functioning soils. species. I know there's a lot of initiatives now where people are essentially isolating different consortia of microorganisms, and they're using that as different inocula in soils that may be oligotrophic or require additional nutrients, right? So those efforts exist. And from the limited studies that we've seen, they're quite effective as well, because if you increase diversity, just sort of the same as the study where I've showed you where rural individuals seem to have much more diverse systems, higher diversity tends to equate to increased stability and better and more resilient and more functional ecosystems. So it is possible to increase it in soils by doing either reciprocal transplants or by essentially applying consortia of different microbes in these new systems. The only issue with it is that depending on how you treat the soil, it will be important to have a recirculation, a process where you naturally allow the soil to regenerate itself. So it's not a long-term process, especially because you are putting in plants that remove all those nutrients. So there will always be a requirement to allow the soil to naturally regenerate itself.
Yeah. Great. And with our gut, is there a quick fix to increase the diversity of our gut biomes?
Yeah, so similar with that, people, there are lots of probiotics. It still remains a bit open whether this is a long-term improvement, because you mentioned stool transplants that have been used for many years in places like China. It's in the literature where people used stool transplants, and that these lead to better health outcome, especially for people that have dysbiosis and leaky guts. Probiotics have been seen as one alternative where people take in lots of probiotics, but looking at whether one particular microbes would increase the diversity, that really remains to be seen, right? Because especially looking at the fact that these are ecosystems, right? Whether they're in soil or whether they're in your guts, microbes rely on each other for interactions. So increasing one does not necessarily mean that you might lead to better outcomes. Often it's more increasing a consortia of certain microbes within your gut that might lead to those improved outcomes.
Right, and on that note, a very diverse diet that will naturally have very diverse biomes is probably our first point of call, and is probably why we're seeing that difference between urban and rural population. There are
yes, from all of these studies, what you can see quite clearly is that a diet that is quite high on fiber includes a different proportion of the various nutrients is always good for you. I mean, it's exactly what your parents told you when you were younger, right? If you eat your veggies, that leads to better health. I mean, and so more fiber is always good.
Yeah. So two more questions in the chat. One says, as you talk about funding, most times our gaze turns to the global north. However, we have resources within the continent which just need to be gathered and focused for such initiatives without the conditionalities of the north. I absolutely agree with that.
Yeah.
Where do we start?
That's true. I mean, I think part of what we would like to do, right, through the African Microbiome Consortia is try more specifically to make links to bodies such as the African Union, to at least lay and make it quite clear in the heads of our policymakers why such studies are important and what are the potential economic outputs from such studies. Because sometimes it's not very clear to someone why you think it's important to look at microbiomes in soil. But by quantifying it, and you can do that by the research where you show increased productivity, for instance, that makes it more crystal clear in their heads, and hopefully that's followed by more investment in this type of research and also in this type of industry.
Ideally, part of our absolutely, ideally, part of our taxes should go towards it because it is fundamental. You've been saying from the start, it underpins the health of our citizenry, the health of our nations. So it shouldn't be a by the way, and we'll do this if we have a bit of money left over. It should be this is the most important thing we're going to be doing.
Exactly.
At the end of the day.
Yeah. But like you say, by continually advocating and sometimes it means saying the same thing over and over in different avenues, maybe it will catch on one day and people will recognize the value and the investment that's required, right? Because what we're calling for means that you obviously have to train a lot more Africans to be able to, one, analyze the genomic data that's produced by looking at African microbiomes. That means, you're putting in a lot more funding in terms of the research that goes with such studies. And that's a considerable investment. And that is possible that those funds can come purely from the continent. But we also have to be strategic, right? There are global funds that are available. What we need to be strategic about is make sure that that investment comes with more advancing our needs and our priorities than it does, people from the global north. So it does mean being a bit more strategic.
Yes. So we'll take two more from the audience. And I have a little one that I'm going to sneak in there. So this is from Dr. Chipstem. He says, you mention and illustrate how greater diversity lies in rural areas. Do you have ideas on how we can increase the diversity of urban microbes and the role that regen ag may play in this process? And how can we demonstrate this importance to consumers? Great question.
Yeah, I mean, so what I did not mention, in that study is that we also took metadata. So we did full diet profiles of these individuals. And what you can see is that in urban areas, you see a greater reliance on fast food outlets. And of course, that means that people are taking and consuming food that is less nutritious and less healthy ultimately for them. And when you look at the people in the rural areas, there's a good mix. A lot of them still rely on subsistence agriculture, planting their own food, making sure that they know essentially everything that they consume. Increasingly now with urbanization, there will be a proportion of people that still go to the formal shops, but they're not consuming as much McDonald's and other chain foods in their diet. So of course that's if we show what effect that has on their gut microbiomes and ultimately the health outcomes, as has been demonstrated from other studies right across the world, that having a diet that is less healthy, low in fiber, is more associated with the development of markers for cancer later on in life. That provides quite clear evidence as to why they need to rely on a much more healthier diet versus one that relies on fast food outlets and so on.
Yeah, but I hear that in the shadows of what we're saying here is we're back to a lot has been done in the developed world and not enough in Africa. So even trying to tell African consumers that this leads to that, well, let's get the science and the data behind it in order to have those conversations. Otherwise, they'll say, but or one could argue that the differences are so great, it's not going to affect us, and we all go off on that train towards fast foods and then crash at the end as well ourselves. But
it's also again making the point of how we should think about our own development, right? Because we've kind of seen what mistakes have been done in the global north, and as much as possible, we want to not repeat some of those mistakes. So everyone relies on the convenience of fast food outlets, but there's a way to regulate the food content and how those fast food outlets, the type of food that they provide. And maybe that's what we should be thinking about.
Absolutely. So we're saying we want better quality food, we want better diversity of food, we want it to be cooked in a healthier way. It can be fast food, but it just has to be better. It shouldn't just be, yeah. Do you think that the resilience on the African continent to COVID-19 was based on our gut biome or anything to do with our biomes? Because there was a big worry that without all of the issues that were in place, the sanitization, the kits, the gloves, the masks, the everything, there was a fear that Africans we were going to die like flies on the continent, and everybody left the continent to go to safer spaces. And we didn't.
Yeah, we didn't. So, I mean, in part, I think part of the answer is the youth in our continent. The diversity almost certainly has something to do with it as well. I don't know whether it's necessarily due to diet, but I'll tell you what, we have one study where we're looking at kids that acquired COVID and presented with fevers. We've got some of the historical stool samples. And part of what we're trying to do now is assess what the differences are in the gut microbiomes of some of those kids, and in order to answer some of those questions as to whether those differences within the gut microbiota and linked to the diets might have led to the better outcomes. So, yeah. But when it comes to COVID, it's quite variable. Yeah, because a lot more of our kids, especially if you look at South Africa, were hospitalized, whereas in other parts of the world, kids seem to fare a lot better when it came to COVID. So, yeah, I mean, there's a lot that we don't understand even from that period, and yeah.
Yeah, I guess, yes. I just want us to say that it was strong.
And I'm careful about saying about the scientific evidence.
Yeah. Okay, so the last question we'll take here, well, we'll take two because I've seen that John has one. He says, I find currently many people taking supplements from outside the country, mostly outside our continent, particularly urban areas. Could this affect the diversity in the short and in the long term? I'm assuming he's saying it affect our gut diversity.
Yeah, that means in terms of supplements, yes. I'm pretty sure that will affect the diversity. But the thing is with the gut, it's also taking probiotics is also a transient solution, right? Because it doesn't have a long-term effect. Even with people who take in stool samples, that will work for a period of time, but it will ultimately change and revert back to its original state. So it does affect it, but whether it has long-term consequences, that is also like an open question.
When you're saying that there's that transient change that might happen, but then it would resort back to normal, I suffer from gluten intolerance. It happened along the way. I've been doing a lot of reading and trying to figure out what it could be. There's a lot of talk about it could be things like the herbicides that are used in our food that affect the microbes that eventually affect our own gut biome, but then affect them in such a way that you can't recover from that. It doesn't matter what you do. They're affected, very deeply, and I don't even know how to explain that. Do you have is there any scientific rigor behind glyphosate and the gut biome?
I'm not aware of any particular studies that have looked particularly at glyphosate and the gut microbiome. But I mean, it makes sense that we know obviously that microbes in any kind of system tend to be impacted by physical-chemical variables, right? What the long-term effect is in terms of that priming your gut microbiota and impacting it long-term, I think that's something that's not quite well understood, but it's foreseeable that it could have an impact. So when I talked about the transient nature is that if what you consume is not good for you, that will of course lead to adverse effects. And when we say normal state, your normal is different from my normal, right? And that's also part of the reason why we need to increase the diversity of studies that look at gut microbiome, because sometimes people have this one outlook that if you take in this probiotic, it will work well for you. But it's not necessarily true for your system, because what defines as your normal gut consortia might be different from someone else, right? It's also an element of the interaction between your own genetics and your own gut microbiota that leads to that outcome.
Yeah, so we're also different, but we're all generalized when it comes to eating and medicine, aren't we? We're all...
Yeah. And I mean, that's why when people talk about, this whole move to have what do they call these tailored medication, precision medicine, part of it depends on knowing exactly what your own gut microbiome is, because that will regulate how different pharmaceutical compounds are metabolized within your gut, right? That's why different people can take the same medication but have different outcomes.
Yeah. So here's our last one. This is from Muritu, who is one of my soil advocates in my current class. Nice question, Muritu. Thank you for being here. He says, thanks, Prof. The generation that destroys the environment is not the generation that pays the price. So he says, what's your 3 words of advice to the common Malinchi? Now, common Malinchi means just generally to us, the people. What are your 3 words of advice? And then 2, are there any plans to train people for free on the basis of your great profession? I know that you're an extremely busy person, so I can almost answer that for you. Listen, we've been talking on email. This is the first time we get to have him sit down in one place. He's an extremely busy guy.
Yeah, in terms of training, maybe I can answer that. I mean, linked to, but you might have missed the opportunity, the ISME meeting that we'll be hosting. We actually got some funds specifically to train African scholars. And these are full cost scholarships where we'll be selecting different researchers from across the continent to come and do a 3-day training program in Cape Town, and then join the ISME meeting, which will happen here as well. So, I mean, those opportunities are there. And what we'll do as well, linked to that training, is that we'll make the event hybrid. So people that can't join in person will be able to join online. So we regularly have such training programs because, like I said, this requires a lot of bioinformatic know-how, and that know-how is currently not available in most parts of the continent, and we're slowly trying to nurture and develop that. So those opportunities will be there. And I think have a look at the ISME 19 website. It's isme19.isme-microbes.org to see those opportunities as they come up. In terms of words of advice, well, yeah, I think you're correct. The generation that destroys the environment is not the generation that pays the price. But it doesn't mean that we, as the generation that's currently doing the destroying, it's not too late for us to change what we do now, right? Because the investments that we make now will also be to the benefit of the subsequent generations. So as equally as, all the bad things we do now will be, felt later, all the good things we do now can also be born experienced by the generations that come after us. So, yeah.
And they will expect it of us. We must be the change we want to see. Absolutely. Good. Well, we've come to the end of our session. It's we've had you here for nearly an hour and a half, and I thank you so much. I know you're extremely busy. I would just like to thank you on behalf of our entire audience here. Thank you for joining us today. We're so honored to have had you share your presentation. We have recorded it, and we will have it on YouTube, and we will now, as the advocates we are, share and share and share, and get more people to see your fantastic work, and hopefully chip in and sponsor your fantastic work and become part of your teams and your consortia, etc. So basically, they'll just be able to reach you at Stellenbosch, and I will leave, end on that note. Thank you very much.
Thank you, Sue, and thanks for the opportunity. And I look forward to working with you as well.
Super, great. We'll all give you a huge round of emoji applause, because that's what we can do at this stage. Thank you, Team iCow. Thank you, everybody who's been watching. And I wish you all a lovely evening and happy Easter.
Yes.
Bye.
Bye.
Bye.